All right, good morning, everybody. We're going to transition into our last session 6, which is marine ecosystem thresholds and regime shifts. We put this session together with Andrew Barton and Jasmine John and myself. Andrew couldn't be here, but Jasmine and I will be moderating the session for these next 4 talks. So Phil Boyd is going to kick us off with phytoplankton regime shifts and climate change.
Is that better? Yes, okay, it's on. Anyway, there's probably not many people know this, but there's a little clause in the small print of OCB which says that the morning after the dinner in the tent, you can sit down if you're a speaker. So I thought, why not? So please excuse me, I'm sitting today, but it's not because of the dinner. So today, I'm going to focus on what's really been an emerging trend in the recent literature. Some papers in the last year or so from Science, from PNAS. I've just cherry-picked a few out. And the reason I'm talking about these in the context of regime shifts is it's probably threefold. First up, I've been doing a fair bit of work the last few years in what you might call the multiple stressor sort of research community, or the global ocean change biology community. And so we've mainly been sort of constrained to sort of laboratory experiments or be at best some sort of mesocosm work that's typified by some of the German labs. And so we want to see what can we learn from this real world sort of data sets and trends. As you can see, we have different data sets here. Here we have one from McMahon's lab on deep sea corals. Going back over the last millennium, stuff from the CPR work here in different regions, and then down here work done at the Cariaco time series, which comes on to sort of the second observation, which is these are looking at important groups from a biogeochemical standpoint, nitrogen fixers, coccolithophorids. So again, we want to have a good look. And then the third observation is I have to ask myself the question when putting this presentation together, are these really even regime shifts? They're certainly not under the classic very rigid definition, which really comes out of both terrestrial and nearshore ecology in the ocean. And so I'm going to also pose the question, are these potentially showing us some sort of rifts around this concept of regime shifts, particularly in planktonic ecosystems, something that we might see increasingly in the future. And so we'll back up a little bit and just, here's the definition. There are many definitions out there on regime shifts. I've seen some pretty aggressive cut and thrust at meetings over the years, people discussing what is and what is not a regime shift. I've highlighted the word persistent here because I think when we look at planktonic systems, they're very complex, there are more permutations, and this notion of persistence may, in the future, have to be modified and made a little bit more subtle. So here's a few examples here from a collation that Scott Doney and I put together a number of years ago. It's been a major growth industry since then. We can see just a quick look at the recent electronic literature. The papers are still coming out thick and fast. So in terms of trying to pick out a reasonably sort of clear-cut example, here's Francisco Chavez's very nice paper from the Equatorial Pacific. In the right-hand panel, we can see the nutrient-impoverished Equatorial Pacific in January '98. And yet, 6 months later, La Nina kicked in. The afterburners of the Cromwell undercurrent have been kind of switched on. We've got lots of micronutrients coming up into the surface ocean. We have this very productive layer snaking out over towards the warm pool. And one of the reasons that Scott and I put this early collation together of regime shifts was, of course, our sort of holy grail in climate change science is to try and get some sort of analog for this. And so we have these two end members. We have really interesting and compelling stuff back in deep time. the Paleocene-Eocene thermal maximum 55 million years ago, gives us the right sort of increases in CO2, the right sorts of warming, the right sorts of shifts in nutrients, but it's way too slow, you know? It's tenfold too slow, and it's the best we have out there. And then we move forward to something like this regime shift, and it flick-flacks in 6 months, so that's a little bit too quick. And so we're still not there yet. However, during the IPCC, we were working on Chapter 6 in ocean systems. We had a pretty good opportunity to poke through the literature, and this was the best we've come up with. This is from the northern North Atlantic, the Norwegian Sea. It's worked by Ken Drinkwater. And you can see it doesn't do too bad a job in terms of both the magnitude and the rate of change. And the nice thing about, you can see in terms of warming, it's about 0.8 of a degree over just over 4 decades, so not too far off RCP 8.5. And the advantage of finding a sort of a scenario like this is when we look at the texture we see here in terms of the ecology, we're not just looking at sort of the base of the food web, we've got zooplankton, fish stocks, phenology, fecundity. So it starts to give us a hint about what might happen, not just at one trophic level, but right through the system. And that's why I think it's worth pursuing this a little bit further. Of course, in this case here, we return back down to baseline, whereas it's going to keep on keeping on, or hopefully not. The other issue we have to face with regime shifts then is what I call this cluster effect. So this is our sort of climate change cluster, the warming, less nutrients, shallower mixed layer, higher CO2, possibly lower trace metals. If we compare that with some of our potential proxies, they have a completely different set of permutations that we look at. So again, we have to be careful in terms of how far we can make a connection with these proxies, because it's not just one thing, it's that whole cluster, in this case for phytoplankton, which is going to concurrently influence what happens in the future. And another interesting paper came out a couple of years ago. Of course, the North Atlantic, through the work done with the continuous plankton recorder, has been a real hotbed for showing us a whole series of interesting trends with respect to regime shifts across the plankton. However, they put this paper together a few years ago where they actually tried to tease apart, using principal component analysis, the relative contributions of climate change in the North Atlantic to climate variability. And here we see that there's not too much of a difference between them. It's around 16% here for climate change explaining the variance, whereas the AMO is explaining still 11% of the variance. And this sort of gets us back to some of the things that Galen and Scott were talking about earlier in OCB about this notion of the emergence, when the sort of climate change signature eventually punches a hole in this envelope of variability, and suddenly we're off on this more sort of unidirectional type trend instead of bouncing around inside the envelope. So again, it suggests with regime shifts that at the minute they are being mediated both by climate variability but also climate change. So again, it makes it difficult to predict and to project how they're going to change in a future ocean. And so just to sort of put that then in terms of the phytoplankton then are going to encounter these range of conditions which reflect both climate change and also climate variability. And that's really where we have to look, because that's what's going to dictate how they respond, and whether they respond in a sort of a plastic way or whether they respond by actually then changing things genetically. And so coming back to these studies then, what sort of trends do we see? Well, in the lower case here, this work at Cariaco suggests evidence of adaptation over a 15-year time series record. That was based on some fairly grunty statistics using max EMT. I talked to a biostats guy at the Gordon conference last week, and he kind of shuddered slightly when I mentioned it, so I didn't take it any further. But anyway, fairly powerful statistics. Some of the work here then from Rivera Calle suggesting, again, that actually secular climate change is actually driving the increase in coccolithophores. The last 100 years, sort of moving into the early Anthropocene, suggests a very clear signature and shift over towards diazotrophs in this record in the subtropical Pacific, and then a range of different shifts that we're seeing in this North Atlantic sort of mixed community. So we're seeing a different range of responses. Perhaps predictably, we're starting to see a little bit of kickback. You probably recognize the PNAS red, red kind of text. There's been quite a bit of cut and thrust, particularly about Andrew Irwin's paper, talking about whether you can see adaptation in 50 years, this is a long enough time series, and so forth, and so on. And so rather than dive into these and really critique them further, I think it's sort of premature to do that. I wanted to see, is there some way that we can move in a way that will complement the statistics, but that actually takes a slightly more maybe mechanistic approach to this, and see, does adaptation at Carriacou make sense, whereas a direct shift through some other means in the North Atlantic make more sense under those circumstances. And so in terms of the issues, you've seen from the slide I showed you from the northern North Atlantic that we see these links and this propagation, that if we see an enhancement of net primary production, typically we tend to see that bumping up through the different trophic levels, perhaps going as far as fisheries. And so with these new examples for these primary producers, it's important to try and learn a bit more. There are some issues. We've seen that with papers by Steph Henson and also Mark Oman, where they say 10 years, 15 years isn't enough. You need the minimum of 60 years, and that's really been based on a partial analysis of the Cal Coffee record versus analysis of the complete record, or in the case of CWIFs, having a look at it and comparing it to some modeling work and saying, no, record's not long enough to actually be able to answer these questions. And of course, the other then issue is that phytoplankton, of course, are basically the ideal tracers in the model. They are, by and large, passive drifters. And so we have this issue of ocean circulation, connectivity to look into. It's a complicated business. And so again, a couple more papers there. One that only came out a couple of weeks ago by Martina Dublin, and then another one that I just heard about last night that Broer pointed out to me. Again, looking at these issues of connectivity and how things are moving around the ocean. And so again, I want to explore some of these notions. And so to do that, again, I'm going to just take you on a slight tangent, if I may. I'll finish up with this slide by Rivero Calle, just to give you one example. So they're using random tree walk models, and they construct 3 of this. As you can see, they've got long laundry lists here. They've got a whole host of things that could be the key drivers for predicting coccolithophore occurrence from the CPR record in the Atlantic. They take 3 different forest models. The first one uses a monologue CO2 to drive just a seasonal variability to oscillate that. The second one then takes a Takahashi climatology for the North Atlantic. And the third one puts both of them together. So this is just a spatial detail that has the seasonal oscillations, and then this puts both. And when we look at these, global PCO2, this king of the hill that captures or explains most of the variance in terms of them predicting coccolithophore occurrence. In this situation, with local PCO2, it does best, but AMO is close behind it. And in this case here, with the Takahashi climatology approach, AMO explains most of it, with CO2 being down the pile. So I'm not going to go into the pros and cons of these 2 approaches and which is the most appropriate, but it's just to say that, as you can see, climate variability and climate change never really seem to be that far away, and so we have to be careful how we look at some of these issues. Now I'm going to take you off on a slight tangent, which is just going to dive very briefly into this multiple stressor literature. And so, of course, moving on from things like warming and ocean acidification, we have a whole screaming scheme of permutations to try and get our head around to work out. We have a lot of biologically influential properties here in the ocean. And so this is sort of, the challenges have been sort of neatly summarized by this cartoon in Nature Climate Change from Rivasil and Gattuso. And so this is kind of the false summit that ocean acidification represents here, sort of with a single driver, single species, and not really looking at things like acclimation or adaptation. They then give us 3 axes that we need to explore, so we need to try and move up this line here from species to ecosystem, and this one here from the single to the multiple driver, and along this one here towards acclimation and adaptation to try and get up into this rarefied air that we really need to get to. And what I'm imagining is that some of the data sets that I've just shown you from these long-term time series are going to be out here in this sort of space. And so the more we can learn from them, the better. And just to illustrate some of the progress we've made then, in terms of moving along this axis here from single to multiple drivers, we can now, through a new factorial design, get up to 5 stressors reasonably easily. So we can look at the whole of the change for phytoplankton. In terms of this axis here, again, there's been quite a few advances in terms of trophodynamics and multiple stressors. And the one that's most pertinent to this talk then is then moving along this axis. This is work where they're taking something like a coccolithophorid and running it for maybe 5 or 6 or 7 or 1,000 generations. And as you'll see in a minute, that's a sort of a relevant time scale with respect to what we're seeing on these longer time scales, with the caveat that these are all done under mean 2100, year 2100 conditions. But again, up to 1,000 different generations. So let's step back to Andrew's paper. He can't be here, but I saw him last week at the Gordon conference. From his work in the North Atlantic, he calculated some what he calls climate velocities. So this is our random plankton drift over a decade. And in terms of the vectors, when he sorts it out, he's got about a 40-kilometer sort of zonal shift. He's got a 10-kilometer meridional shift per decade. And what you can do with that then is we can start to back that out if we assumed 150-day growth season, give them a modest growth rate. That gives us around 750 generations of phytoplankton over a decade, which we can then relate to these sort of climate velocities. We're not really sure about overwintering, but what that allows us to think about is what's the scope for physiological change over 750 decades. We've seen from these experimental evolutionary biologists that they are seeing changes over that time, but of course they're not looking at things under fluctuating conditions. So how might we do that? I've just overlaid this drift pattern on a number of different scenarios. Here's one from Marina talk the other day. What happens if we have this very variegated sub-mesoscale pattern? That's really going to influence the sort of conditions that the phytoplankton encounter versus a more graduated sort of situation here, and one of which we might see something a bit more like an abrupt gradient. And depending on what sort of gradient you see, you're going to see the phytoplankton are going to encounter a very different range of conditions over this decadal trajectory, when they're dividing for some groups primarily asexually with the occasional need for sexual division. How might we get at this? Well, this is some fairly new data from a geotraces process study. This is, you can see across the sub-mesoscale here, we have a wide range of different conditions. This is in the center of an eddy. And you can see that we've come up with a whole series of different maps of different properties. Dissolved iron, nitrate, silicate, ammonium phosphate, and some other biological properties here. So we can start to build up a little bit of a picture of the sorts of permutations of conditions that a phytoplankton might see as it moves around this course. The next thing we have to do then is take some of these plots. These are CO2 affinity plots from the Hutchins lab. These are some of your standard reaction norm plots. And if you imagine we get those data, we can move our little red ball up and down these curves, and we can actually see, are the conditions that are being encountered really making any sort of difference at all? You know, are they pulling them up here, or are they always maxed out, or are they falling off the cliff and things like that, which gives us some sort of hint about what might happen. Because there's some key issues here, and coming back to this issue of the emergence that Galen and Scott talked about, I placed the emergence squarely in here, and so, you know, we have these issues where, you know, organisms when faced with change can get out of Dodge City, or they can die, or they can somehow adjust, and then adjustment might be initially using their innate plasticity, their phenotypic plasticity, with eventually some change here, some genetic change where they actually start to have directional selection. And so we're suggesting that the emergence might be a key inflection point where this actually takes place, because while you're still bouncing around in this envelope of variability, you're not really getting unidirectional change, and it's really unidirectional change that you need to really start to enable directional selection to take place. And so you can see, as we've alluded to in this recent paper, this is sort of a very messy area. When you look at people who work on guppies and all sorts of other things who've been doing this work for years, there are all sorts of issues about, will phenotypic plasticity evolve, the role of epigenetics. I'm not going to go there in this instance, but what I'm saying is that over these long timescale records that we're seeing being published now, 60 years, 100 years, already these guys hitting the emergence. And so we've done a little bit of work on this. Scott and Ivan did a bunch of model runs. The CESM 1 RCP 8.5 run for the polar Southern Ocean. So these are all the biologically influential properties here. All the blue dots are the monthly anomalies across the rate of change as we move out to 2100. Rather than you go stir crazy looking at the blue dots, I've summarized them here. This is a league table of the emergence times. So CO2, because it's driving the show, it should emerge within a decade in the Southern Ocean. But then as you look through salinity, ice fraction, warming, it may be almost 4 decades before it emerges, and so forth and so on. We also see increased variability as we move out to 2100. So let's just jump to the next slide and get away from the tyranny of blue dots. And what we see then is that with this shorter emergence time for CO2, it's managed to jump across this inflection point. So suddenly, for the cell, it can adapt to a change in one property, whereas for these other properties, it's still acclimating to them. So that I'm not quite sure how that works out in terms of energy allocation or things like that, but it looks a bit like, physiologically speaking, you're doing the splits. It's kind of a tricky one. And so you can imagine then if we look another 20 or 30 years' time, then salinity and sea ice and temperature would have jumped across this gap here. So again, you're getting a range of different, getting an approach where you can adapt to some of these things, but you're still having to acclimate to them. So it starts to make this thing kind of messy. And so just to summarize that part of the presentation, it's looking like we're going to see these complex strategies that are going to be needed, in which things are going to have to actually physiologically straddle, both including acclimation to things, but also adaptation to things. And as I say, CO2 is the one that's really driving this show. It's going to be the first one to emerge, and so we're going to see probably adaptation to that. So, as I say, these are things I think that are all in this mix that, in addition to using high-powered statistics, we have to start to think a little bit outside the square. Just to conclude then, regime shifts, certainly in planktonic ecosystems, as compared with benthic systems or systems in the near shore where things just flick-flack from one to the other. The little M&M rolls up to the ridge and then falls down the other side and gets stuck in the well and things like that. I think in the open ocean, things are going to be a bit more nuanced than that. And we're going to see a whole raft of different possibilities and permutations. These recent studies are of great interest to us because I think they represent a tremendous resource. But I'd hate that resource to be sort of stymied by just saying we've thrown the kitchen sink at them statistically and take it or leave it sort of thing. For example, it intrigues me that in the mapping paper in the subtropical Pacific, that as soon as the Industrial Revolution starts, the azeotrophs have come in because, the world was a very different place then. There was a lot less anthropogenic CO2 knocking around, but yet the azeotrophs they're in already. I found that sensitivity to be incredible. And so there are lots of exciting questions to discuss, but I don't think now is the time for people to kind of slam dunk these papers. It's time to have a discussion and try and broaden it out with what we're also learning in a mechanistic sense to explore this. And as I say, so really it's a plea that we want to try and recast some of these data sets and say, well, based on the range of environmental variants that you're seeing, would we expect us to move towards coccolithophorids or towards diazotropes? And finally, I think the other thing that's really emerging, it really jumped out at us last week as a consensus thing at the Gordon conference, was that by focusing a great deal of modeling attention on how the mean is going to change in the future, we really need to play a bit of catch up with the variance, because I think we think that the variance could be just as important, just as influential in shaping what happens with marine biota in the future. And I'll leave it there. Thanks.
Do we have a few minutes for questions?
I have a question. That was a really thought-provoking talk, and I'm just wondering with some of those predictions of timescales of seeing some of this adaptation versus acclimation and sort of that space, if you've done any modeling or simulation in parts of the ocean ecosystem that are highly variable. We've been hearing a lot of talk, in the Indian Ocean, this really dynamic system versus the oligotrophic ocean, and do those time scales change just by that background variability?
Yeah, no, to say it's very much a fledgling field. We had a lot of high-powered people from the experimental evolutionary biology community there last week, and there were a lot of blank faces, and that's a good question. And I think it's this whole issue of this as I tried to show in that figure, this very fuzzy area in the middle, where we're really not sure what the connection is. You know, as I say, for now we've just plonked the emergence in there and put it up there and said, is this a putative transition point? But we don't really know. But it's good to put it out there anyway to start some discussion around that. So, yeah, sorry, Kathleen.
Phil, do you think that there are things that we need, additional things that we need to be measuring in the future in terms of our time series programs, things that, you know, the data that we collect that we use to measure these sorts of regime shifts that we're not doing now? I'm thinking of things like ways of measuring, I guess, physiology, or what do we need to do in the future to get at some of these mechanisms that we might not be doing now?
Well, first up, Debbie, I'm surprised you're looking for more, because having seen the export spreadsheet, I just thought about an America's Cup spinnaker. and so if you want,
there's always something else, Phil.
That's true. I mean, a number of years ago, Scott and myself, we had a paper, and we put some scenarios in there as a way that you might be able to look at what you were measuring in the time series, and that included nothing. This is just for phytoplankton. Nothing changed, but everyone's physiological rates changed. That's really tough for a time series. Or you have a situation where you had floristic shifts, and you know that's going to be pretty conspicuous. And then you maybe have a change where you just didn't expect it, and so again, it's hard to go for that. I think once you start to move out into the physiology, it does start to get very tricky in terms of what to pick up on. But certainly, I think it would be prudent to archive a bunch of samples and just tuck them away somewhere for next-gen sequencing or whatever's coming around the corner. I think just having a black box set of samples would be a very wise investment. You know, I mean, we tried to do something like that during the biological geotraces component. We didn't quite pull it off, but I think that would be that would be something very valuable to have. In the same way, sometimes you get an email saying, we just got some samples from GSX, could anyone use them? You know, having archived material sometimes really is a great legacy. So.
Thanks for a really great talk. I'm coming to the question that I asked on the first day, and it's actually banging my own drum here. It's a question about mixotrophs, and you spoke about physiological plasticity. Most of the models that we have do not have mixotrophs, and I wonder that would be a direction that we need to explore in the near future.
We had Kevin Flynn at the Gordon conference last week, who Holly name-checked a number of times in a talk, and a couple of people sort of asked the question, well, you know, why don't mixotrophs, you know, take over the world? And Holly also alluded to that, and of course, by using the trade-offs with traits. I mean, if you have mixotrophy, you can't just flick-flack at will. You know, you have to be able to spin up different enzymes and change systems. There's transcription and translational process to get through. So you can't just do that in a matter of seconds. And so some modelers have been able to use that successfully to put some bounds on it. And so yes, certainly there is some plasticity there. But again, it's really sorting out those traits, and probably using some sort of dynamic energy budget would be a way to start to delve into that further.
All right, let's take Phil one more time.